So last time we talked about the splicing of pre-mRNA to form post-mRNA by removing the introns and ligase enzymes binding the exons together. We talked about how this is controlled by the intron-exon junction, and we talked about how one of the mutations that can be responsible for muscular dystrophy are a mutation at this splice site, so the TTAG region on either side of the intron, which results in that intron not being spliced in this step and resulting in production of a dysfunctional protein. Another example of a splice site mutation creating a dysfunctional protein is the case of thalassemia, which is where red blood cells in the patient are distorted and immature, containing a nucleus, while red blood cells are typically enucleate, which gives them their biconcave shape. So if you look at the image on the bottom right, it shows what blood cells look like in thalassemia instead of, I'm sure you saw normal healthy red blood cells where they have kind of an hourglass kind of shape because they do not have a nucleus. So what is this mutation doing? So it's a point mutation at the splice site of the beta globin gene, resulting in one of the introns not being removed. It's caused by a single base pair mutation, which we call a point mutation, and this mutation occurs within the intron-exon junction, resulting in abnormal splicing, so an intron is not removed because of this point mutation, and leading to a dysfunctional beta globin protein. This dysfunctional beta globin protein causes the odd characteristics of thalassemia and causes the symptoms of anemia, and we can treat it via activation of fetal hemoglobin genes because the mutated version of the adult globin gene, which is causing the thalassemia, that's the adult red blood cell globin gene. We can activate the fetal hemoglobin gene, which switches itself off as we get older, and if we turn it back on, then we can produce healthy hemoglobin and healthy red blood cells. This is the same way that sickle cell anemia can be treated, but it's very new and it's very costly, so it's not really a practical cure right now. We can only really treat the symptoms of anemia. More on alternate exon splicing. So actually, I don't know if we covered that last time. So basically, a pre-mRNA may mature into mRNAs with alternative structures depending on how the exons are spliced together. So if we look here, we can see that there are multiple exons. In a regular gene, there would be many exons. They can be kind of mix-matched together like a jigsaw to produce alternative mRNAs depending on how they're spliced together. So that's all that alternative exon splicing really is. It will alter the gene product because obviously different exons in different orders in the post-mRNA is going to produce different polypeptides when it gets translated. There are several instances of this altering phenotypes. So, for example, alternative exon splicing is responsible for sex determination in flies. Alternative splicing also means a single gene can produce a related set of mRNAs that code for a small family of related proteins. And these related proteins are called protein isoforms. If you've done any immunology stuff, you'll know it's kind of the same terminology for immunoglobulin molecules, which have various different isoforms like immunoglobulin G, immunoglobulin M, etc. So it basically just allows a single gene to produce a variety of different proteins that can serve different functions. And this is the norm for creating protein diversity in eukaryotes. How is exon shuffling different to alternative exon splicing? Well, basically, alternative splicing happens to the mRNA. So it's happening in this step. Alternative exon splicing, where you have this pre-mRNA shown on the right, it's having its introns removed by the spliceosome, recognizing the intron-exon junctions. You're having the intron separated. The intron folds into the circular lariat structure, and then ligase enzymes are going to come along, and they're going to basically glue the exons together in whatever order that we need for whatever protein we're making. So if we think about like this example, it may glue the exon on the right to the 5' end on the left of the exon on the left, or it may glue them together as they're shown here. It just means that when this post-mRNA is going to be translated at the ribosome, it's going to produce functionally different polypeptides, which are going to produce functionally different proteins, which we call protein isoforms, which are going to serve different functions. So that's alternative splicing. But exon shuffling is happening inside the genome and not on the mRNA. So it's a similar concept, except the exons are being rearranged within a gene, or exons may be transferred between genes via genetic reintegration. So alternative splicing happening at the mRNA, exon shuffling happening within the genome. The proteins produced through exon shuffling are called mosaic proteins, which basically just means the proteins are coded for by exons that originally came from different genes. We talked before in the previous video, or the one before that, about how prokaryotes lack introns while eukaryotes have introns, and it's partially because there's strong selective pressure against junk DNA in smaller organisms that produce large amounts of offspring through binary fission, like prokaryotes. And there are two main theories as to why we have splicing in introns in eukaryotes, but not prokaryotes. The first one is the intron early theory, which suggests that an early ancestor of both groups, the prokaryotes and eukaryotes, already had a spliced genome, and then prokaryotes later lost the spliced genome and the introns. And then the intron late theory, which suggests that the early ancestor was unspliced, and then prokaryotes retained this unspliced feature, and then intron splicing is a derived trait of the eukaryote group. The intron late theory seems to be more credible when we think about the selective pressures acting on the organisms, because we would expect the ancestor to also be very small to produce large amounts of offspring, so it would have selection pressures against junk DNA, and less genetic drift would be less powerful, natural selection more powerful. So we'd expect the ancestor to also have an unspliced genome, and then as eukaryotes got larger and more case selected, they had larger genomes with more junk DNA. So that's all for that, thank you.